Thank you very much for the introduction, and thank you all for being here today. And I'd also like to express my appreciation to the Azrieli Foundation for this amazing opportunity to speak with you all today. So my name is Anand Chopra, and I did my PhD in Canada, and now I'm a postdoctoral fellow at Ben-Gurion University of the Negev in Beersheba, Israel. So today I will be talking to you about proteins, and specifically a protein called SETD6 and how this protein is involved in controlling the cell biology in certain aspects. So for anyone outside of cell biology, the concept of how proteins can be connected to the behavior of a cell can seem quite abstract in nature. You might think of proteins and think of nutrition, like chicken. But actually, the way I see proteins, they're like people. So as we can see over here, in this sketch of a city street, we can see there's multiple jobs that are taking place here. We can see there's a taxi driver involved in transportation. There's people involved in disposal of garbage. There's people involved in signaling events that are happening in the city through selling the newspaper. Similar to a city, proteins are people with distinct jobs that are essential for the city to run. A common example of a protein with a unique job is hemoglobin, whose job is to transport oxygen from the lungs throughout the body to adjacent tissues. However, as I mentioned, I'm specifically interested in a protein called SETD6. So who is SETD6? What job does it perform? And how is it important in the cell? SETD6 is shown here on the right-hand side. It's this very intricate structure. And what it does is its job is very specific in that it will chemically modify other proteins. It will modify a piece of a protein called a lysine, as shown here, and it will do this by adding what's called methyl groups to them, as shown in red. Now, this sounds like a very specific role, but there's multiple layers of complexity here. So how can we define the role of SETD6 in a larger scheme? We can say that SETD6 is a commander of other proteins. So for example, SETD6 will modify a protein, shown here in orange, by adding a methyl group. And this methyl group is akin to being passed out a note with specific instructions for this protein to execute its job. So the methyl group will thereby change the function of the protein and then affect relevant cell biological processes. So in the Levy lab, what we're interested in is all three aspects of this. One, who is SETD6 passing out notes to in the city, meaning what are the proteins which are modified by SETD6? What do these notes actually say? Meaning, what is the effect of the methylation on protein function? And three, how does this affect the city as a whole? Meaning, how does this methylation event affect cell biology? So going back to the first layer of this, we need to first understand who SETD6 is meeting in the city and passing notes to. Now, in the cell, there are about 20,000 genes which are coding for proteins, and the possible number of proteins in the cell are estimated to be between 80,000 to 400,000. So this presents a challenge. Fortunately, there are many ways we can take to identify which proteins are being methylated by SETD6. We have methods which are cell-based approaches. We have in vitro approaches, which take place in a test tube, and we have combinations of the two. One highly efficient approach, which we have taken in the lab, is to use these high throughput technologies, which are called protoarrays. And what these are essentially these chips which have spotted on them individual proteins, which are spotted onto about 10,000 spots. And what we can do is monitor which spots SETD6 is able to add a methyl group to with the use of radioactive labels or antibodies. So after we've identified who SETD6 is passing out notes to, we can then move to understand what do these notes say, as well as how is this affecting the city as a whole? How does it impact cell biology? But the answers to this question are very contextual in nature. So for example, SETD6 is known to modify a protein called P65. And P65 is a gatekeeper of inflammation in the cell. However, when SETD6 passes a note to P65, this tells the protein to do its job less, and then we see less inflammatory response as well as less proliferation in the context of osteosarcoma. So this is an example whereby SETD6 is inhibiting the properties of a specific type of cancer. But we can also have the opposite case, for example, where SETD6 can modify a protein called twist, and this can enhance the cancerous features of glioma, which is cancer of the brain. Another very visual example of how SETD6 can influence the cell biology through the methylation of proteins is its ability to control the function of a protein called PAC4. Now, PAC4 is a protein involved in cell adhesion, the ability of cells to adhere to a surface. As shown on the right here, we have an image of a cell where we have conditions that are permitting SETD6 to methylate PAC4, and on the zoom in, we can see that the surface of the cell is quite smooth in nature. However, when we manipulate the cell to force conditions where we have no methylation of PAC4, we can see the morphology of the cell changes quite a bit, and we can see that the edge of the cell, the surface, have these spikes or these spikes which come out, and these are called actin structures, and they're needed for the cell to adhere to surfaces. Besides affecting the ability of cells to adhere to a surface through PAC4 methylation, it can also affect the ability of cells to migrate over time as well. So we can track the movement of these cells for a period of about 4 hours and see how they move. So on the right, we have conditions where we have methylation of PAC4, and over the course of 4 hours, we see the movement of the cell is quite limited in nature. However, when we force conditions of no methylation of PAC4, we can see the cell is able to move quite freely throughout the dish. So I've provided you with just a few examples of how SETD6 can control other proteins and thereby affect cell biology. One example is showing the ability of SETD6 to affect cell adhesion. One example is showing its ability to repress cancerous features through methylation of P65. And then we have an example where SETD6 is inhibiting, SETD6 is preventing cancerous properties through its methylation of twist. But SETD6 is also involved in other processes as well, such as the ability of cells to divide, the ability of cells to make other proteins known as translation. So what I want you to take from today is that SETD6, its role in the cell is highly contextual in nature, whereby it will control cell biological functions, specifically through the substrate it will methylate. And with that, I'll be happy to take any questions or suggestions. Thank you.